So mass spectrometry is an incredibly sensitive tool. NMR is a fantastic tool for analyzing molecules and identifying molecules, but you need a lot of material to take an NMR spectrum. A mass spectrometer, because you're looking at ions and you can detect ions incredibly sensitive, you can detect individual ions essentially. By doing spectroscopy inside a mass spectrometer, we can measure well-resolved infrared spectra with the sensitivity of mass spectrometry. And that's what makes it really unique, I think.
Welcome to the Smart Biotech Scientist. I'm your host, David Brulman. Today's guest is Professor Tom Rizzo, who is the former Dean of the School of Basic Sciences at EPFL in Lausanne, Switzerland, and now Chief Scientific Officer at Isospec Analytics. We're diving into cryogenic infrared ion spectroscopy, a game-changing analytical technique that could revolutionize how you identify unknowns, characterize glycans, and solve those frustrating structural puzzles in your bioprocess development. If you've ever struggled with molecular identification using traditional mass spec alone, this conversation will open your eyes to what's possible today. Let's get started. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brulman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Tom. It's good to have you on today.
Well, thanks, David, for having me. I've listened to a few of your podcasts and really enjoyed them, so it's an honor to have the opportunity to be on.
Absolutely, it's a pleasure. Tom, share something that you believe about biomolecular analysis that most people disagree with.
Like to be too controversial, but I would say there's been no fundamentally new commercially available technology for the analysis of biological molecules in the last 10 years, probably since the introduction of ion mobility into mass spectrometry.
All right, and we get more into the nitty-gritty of analytics. I'm really excited to unpack that. But first of all, I would love to dive into your story, Tom, because I'd love to hear what got you started in the world of science, what sparked your passion for chemistry, and what were some pivotal moments throughout your long and outstanding academic career.
This will date me a little bit, David, but I grew up in the 60s, and I grew up in New York. In 1964-65, there was a New York World's Fair that wasn't too far from where we lived. So my parents would take us like every other weekend to this World's Fair. And all of the big technology companies had these buildings that were with presentations and demonstrations. There was IBM, there was Bell Labs, there was DuPont. And I was just fascinated by all of this. And I remember very, very distinctly the DuPont exhibit where they did chemistry demonstrations. And I was fascinated by this. In fact, many years later, I did those same demonstrations in front of my chemistry class at university, even in my kids' elementary school classes. So this kind of sparked my interest in science. And so I knew I wanted to be a scientist, but I wasn't sure whether it was going to be chemistry or not. I thought, in fact, I would be a physicist. I thought my mathematics background was pretty good, so I was planning to go into physics. But I took an advanced placement course in chemistry my last year in high school. And part of the course, the teacher would give us these little vials with unknown samples, and we had to determine what was in it. This was really a start of my interest in analytical chemistry, if you will. And the way we were graded on this was that she would give you a piece of paper with tabs on them, and the tabs had different answers. And if you pulled the first tab and got it right, you got the highest grade. If it took 2 pulls to get it, then your grade went successively lower. So I set up a little laboratory in my room at home to do these chemical tests. So that really grabbed my interest and sparked my interest in chemistry. So I decided to do chemistry at university, but I was oriented more towards physical side of chemistry. And so in the end, when I went on for a PhD, I pursued physical chemistry, but I did a minor in physics as well. So I've always kind of kept that interest in physics. But deep down, this sort of interest in analytical chemistry was there, I think, back from the early days. So when I started my PhD, I was doing something very physicsy in a sense, right? I was really looking at the physics of molecules by how they interacted with light. So the whole field of spectroscopy was fascinating to me. I really liked the tools of physics. I thought lasers were cool. That's what I pursued. But by the time I finished my PhD, I asked myself the question, how large a molecule could we get interesting information from by using spectroscopy? So I went to do a postdoc at the University of Chicago with Don Levy, a distinguished professor, who developed some techniques to put large molecules into the gas phase and measure their spectra. I did that, and in fact, we measured the first spectrum of an isolated amino acid in the gas phase at low temperatures, and that was kind of my start. So when I went on to my first faculty position at the University of Rochester, this was my goal. I had decided to combine mass spectrometry and laser spectroscopy as a new way to get information about biological molecules. But I had no reputation in the field of mass spectrometry whatsoever, and I was trying to get my grants funded, and no one wanted to. I got close at the National Institutes of Health and close to the National Science Foundation, but no one wanted to fund this kind of thing. Took a step back for I had to get tenure as an assistant professor. So I took a step back and went back into looking at more the physics of smaller molecules. And I managed to do well enough to get tenure. But then shortly after I got tenure, I was approached about taking a position at the École Polytechnique Fédérale in Lausanne. And I was asked to apply for this position, which I did, and I got the position. And so we packed up our family and moved to Switzerland, where I continued my work looking at sort of the chemical physics of laser-excited molecules. But I always had in the back of my mind, there's these experiments I wanted to do as an assistant professor, combining laser spectroscopy and mass spectrometry. I never could get the funding to do it. And so an opportunity came at the EPFL in Lausanne, where I had spent 2 years as a department head. I said, I'll never do this job again. It was really a totally uninteresting job. But the president of the institution changed, and he asked me to take back over as department head in chemistry. I said I'll do it under one condition. I've always wanted to do this experiment using laser spectroscopy and mass spectrometry together, but I never could find the money to do the project. This was in the year 2000. And so I said, if you give me the money to build this machine, I'll serve another term as department head. So he did. He asked me to write up a project, and I obtained 400,000 Swiss francs to build a new machine. And that was the beginning of the end for me, in the sense that I've done that ever since. I've been involved in the spectroscopy of biological molecules in combination with mass spectrometry.
Wow, what a fascinating story, and I love your persistence throughout your story. And just a little fun fact, Tom was my professor at EPFL. I'm really happy to reconnect, and I still remember your physical chemistry class, which I liked a lot. And there is one thing that stays with me till today, the umbrella flip of the ammonia molecule in quantum physics, which was a pretty good class. So I still, 20 years after, I think it's been about 20 years-ish, I still remember that part of the class. Throughout your career you recently finished your career at the EPFL, and what led you now to becoming the Chief Scientific Officer of Isospec instead of enjoying your retirement life? Like, how did this come about?
Good question. As you approach retirement, you kind of look back on what you've done and you say, what has it been good for? And most of what I did during my career was curiosity-driven research. I was just interested in what can we learn about large molecules from spectroscopy at low temperatures. But early on, I wasn't thinking particularly practically about it, making a product or anything. But sort of towards the end, kind of think about what is your legacy in science going to be? I developed this passion for wanting to see the techniques that we've developed in the laboratory really make it into practice, make it into a commercial instrument that people could use for biomolecular analysis to help diagnose disease, for example. And so that really became a passion of mine. And I had a couple of postdocs in my group who were equally passionate about that. And so about a year before my formal retirement from EPFL, we started this company, Isospec Analytics, and I signed on as chief scientific officer. And so then when I retired a year later, I kept my affiliation with Isospec and left EPFL, which in fact was a great way to retire, because when you retire from an institution after 30 years, from one day to the next, you go from being somebody to being nobody, right? But at company, I really felt like my expertise and my experience is really highly valued. It's a great way to transition out of the university. And the world of startups is a completely different world from the academic world. And so there were so many new things for me to learn. I found it very stimulating.
Before we continue, biotech founders, I want to talk directly to you. Do you know that feeling when an investor asks about your manufacturing strategy and you freeze? One wrong CMC decision can cost you $2 million and 18 plus months of delays. Yet many CEOs are flying blind on these critical decisions. That's why I created the 1-week CMC strategy assessment. In 1 week, you get a personalized assessment of your molecule and development stage. We identify critical gaps before they become million-dollar mistakes, plus a strategic roadmap for high-stakes investor meetings. One CEO told me, David took me from feeling powerless about our CMC roadmap to confidently pitching investors. An absolute game changer for time-pressed CEOs. Whether you are preparing for Series A, approaching IND filing, or facing scale-up challenges, this gives you confidence to ace investor and board conversations instead of surviving them. Ready to stop second-guessing your CMC strategy? Check the link in the show notes and secure your spot. Because your breakthrough therapy deserves a solid CMC strategy. Now, let's get back to our episode. So let's unpack the technology and let's dive into the nitty-gritty of what you're developing, because you are developing a novel infrared spectroscopy method, but this has been the cornerstone of analytical technique for a long time, for several decades. So can you tell us what fundamental breakthroughs you have developed or you're further developing, how your method is different, and how is it different from the traditional IR approaches?
There are several really important differences. You're right. Infrared spectroscopy has been around for decades, and it has been a cornerstone, but there are several drawbacks in traditional infrared spectroscopy, particularly if we're talking about in the condensed phase. Gas phase infrared spectroscopy can be extremely detailed and precise because the molecules are isolated. But for biological molecules, for example, where they're in solution or some kind of matrix, the spectra tend to be broadened by their environment. So molecular spectra, infrared spectra in solution, if we're talking about vibrational spectra, the vibrational modes of molecules are affected by their environment. And so if they're in solution, then the bands become broad and then less distinctly characteristic of a particular molecule. If one could pull these large molecules, like peptides or sugars, into the gas phase and remove them from their solution phase environment, first of all, the spectra become much simpler. The lines become sharper, And then the second part of it is that if you do this at cryogenic temperatures, the spectra becomes simpler still because bands become narrower. And so one can do cryogenic spectroscopy in matrices, for example, and one can get very sharp spectra if you put things into rare gas matrices. But that's a pretty laborious procedure. And to do that as an analytical tool, it doesn't seem so practical. So if we could put molecules in the gas phase and then cool them down to cryogenic temperatures, you have the simplification of low temperatures, but in an analytically accessible way. But perhaps the most important aspect of how we do it is we can measure the infrared spectroscopy with the sensitivity of mass spectrometry. So mass spectrometry is an incredibly sensitive tool. NMR is a fantastic tool for analyzing molecules and identifying molecules, but you need a lot of material to take an NMR spectrum. In a mass spectrometer, because you're looking at ions and you can detect ions incredibly sensitive, you can detect individual ions essentially. By doing spectroscopy inside a mass spectrometer, we can measure well-resolved infrared spectra with the sensitivity of mass spectrometry. And that's what makes it really unique, I think.
So just to fully understand that, your method is not using mass spec, or is it also coupled with a mass spec?
It's actually performed inside a mass spectrometer. So we put a cryogenic cell inside our mass spectrometer. We can separate our ions using liquid chromatography or ion mobility or whatever you'd like, any of the techniques that couple to mass spectrometry. Once those ions are separated, we put them in a cryogenic cell. We measure their infrared spectrum. Molecules that are mass selected, they could be mobility selected, they could be selected by liquid chromatography. So it's all done inside a mass spectrometer.
Oh, I see. And can you just recap what are the main advantages over the traditional MS-based methods? Is that on the sample matrix level, the quantity, or what are differences there?
Okay, so MS is used very widely for identification of molecules, and it's a very effective technique. However, it has some drawbacks. So, for example, when you have different isomers, molecules that have the same mass, and they may only differ very, very slightly. So the position of hydroxyl group on a molecule, for example, or in the case of sugars, which have huge problems with isomeric complexity, distinguishing just the linkage between two sugars, for example, is very, very difficult to determine. Molecules that are so similar like that can fragment similarly. And so it can be difficult to distinguish isomers by their MS spectra. Now, there's been an interesting paper within the last year by a group in Nijmegen. So often to interpret MS spectra, you use calculations of what the fragmentation products would be. And they actually compared the predictions of these calculations with measurements of fragments that they made using spectroscopy. And they found in a large majority of the cases, the calculations were just wrong. It's just too difficult to calculate accurately the fragmentation patterns of molecules to be sure of an assignment using MS techniques. By contrast, the infrared spectrum of a molecule that's cooled to very low temperatures is an absolute distinguishing fingerprint of that molecule. No two molecules will have exactly the same spectrum if you measure that spectrum at high enough resolution. And so a cryogenic infrared spectrum using the technique we call CIRIS, which is cryogenic infrared ion spectroscopy, an infrared spectrum measured like that can tell the difference of simply an OH group that's pointing up or down. That's enough to change the vibrational frequencies of a molecule and distinguish one isomer from another. And so it's very, very sensitive, but also it gives you a high degree of certainty in your identification. So it's a new data dimension. You asked this initial question about this controversial statement about biomolecular analysis. I said there's no new technologies. What I meant was there really no new data dimension in which to analyze these molecules. But a cryogenic infrared spectrum adds a new dimension to the measurement. So in addition to getting a retention time and to getting its mass and getting its fragmentation pattern, you now just think about pulling this out in a different direction for every mass. You now get a cryogenic infrared spectrum, which is like a barcode or a fingerprint of that molecule. It's really a new dimension to biomolecular analysis.
So in layman's terms, it's almost an orthogonal method, getting another dimension of your whole spectrum because you have traditional mass spec data as well in addition to the infrared.
Exactly. That's exactly right. We get all the information that traditional methods do, plus this new dimension. And we can do that fast enough that it really doesn't increase the measurement time. We've really optimized this technique so that in the same amount of time, you get a completely new dimension of information.
And to what extent does this new technology integrate in traditional workflows? Because a lot of biotech companies work with LC-MS setups and so on. Would that be compatible or would that disrupt the workflow?
If I can just digress for a moment, one thing I've learned going into the industrial world or the business world is that it's not just good enough to have the best ideas or best technique, because there are certain ways that people work, and to change the ways that people work is not so easy. If you've been doing something a certain way for years, to go into a pharmaceutical company and tell people, oh, okay, you now have to do things differently, that's a pretty tough thing to do, right? So you want to be able to integrate these things as seamlessly as possible. Ultimately, and there's not yet a commercial instrument employing our techniques, although we're in a collaboration with Agilent at the moment to introduce our technology into a commercial instrument. So we're currently doing that. We have our own instrument that we've purposely built in our laboratory that we use for analysis. But the idea is that you would be able to follow your current workflow. That is, you do your LC-MS, and inside the box of your mass spectrometer, there would be a laser which would scan as your peaks elute from the LC and go into your mass spec, and you analyze the mass. At the same time, you'll measure an infrared spectrum. And so really the perturbation to the workflow would be almost hidden. You wouldn't see it, except that up on your screen, you would have this new plot of infrared spectra of these species. So that is our goal, to make this as perturbing the current workflows as little as possible. And with today's laser technology, when I started as a PhD student, lasers were big, complicated things. You needed to almost have a degree in physics to be able to tweak them and to get them to work. So the lasers will be in the box of your mass spectrometer. You won't see it. There won't be anything to adjust. So it'll be totally transparent. And so hopefully it will not impede current workflows. It will just add this new data dimension to their analysis.
That's it for part 1 with Tom Rizzo. We explored the fundamental science behind cryogenic infrared ion spectroscopy and how it addresses real analytical challenges you face in the lab. In part 2, we'll dig into practical applications for biomarker discovery, therapeutic development, and how this technology is becoming accessible for biotech companies like yours. If you found this valuable, please leave us a review on Apple Podcasts or wherever you're listening from. See you next time. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.